Hey guys, I hope you guys are doing well. Today, I'm gonna talk about. My name is Ari. Today, I'm gonna talk about meiosis. If you guys don't know, there's two types of these processes. There's meiosis and there's mitosis. But today, we're gonna talk about meiosis. So, meiosis is known as a process where a single cell divides twice to produce four cells containing half the amount of the original generic information. That means like the mom and the dad. So meiosis, in other words, is like generic variety. It doesn't make any body cells. It doesn't make any cell at all. It's just like the female. In the female, it creates the eggs, and in the male, it creates the sperm. Basically, meiosis is the reason why two siblings from the same parent can look totally different, but like similar in the same way, because it takes half the amount of the genetic of the dad and then half the amount of the genetic of the mom, and then they combine and then they make the kid. But it's kind of hard to understand, but I do understand it. Like, it's like when you see a family of like four kids and they all totally look like totally different, and you'll be like, but how they look so different? But that's why meiosis is the process where you can see kids from the same parents and they look totally different. That's meiosis. So there's two phases of meiosis. There's meiosis 1 and meiosis 2. I'm gonna talk to you right now about that. So there's prophase 1, and prophase 1 is when the chromosomes condense and the nuclear envelope breaks down. The second step is the metaphase 2, and is the pairs of homologous chromosomes move to the equator of the cell. So it's like steps. The anaphase 1 is when the homologous chromosomes move to the opposite pole of the cell. And the last one is telophase 1. It is where the chromosomes gather all the poles of the cell and the cytoplasm divides. And there's also the second phase, that was the metaphase 1, and now we go to the meiosis 2. So prophase 2 is where a new spindle forms around the chromosome. It goes on. Metaphase 2 is where the chromosome lines up at the equator. The anaphase 2 is when the centromeres divides and the chromatides move to the opposite pole of the cell. And then the telophase 2 is a nuclear envelope forms around each set of chromosomes and the cytoplasm divides as well. This is such a nice process. So I have this cute picture where you can see it's not the same. They're kind of the same as the work we did that was due today as well. And you can see the picture. You see prophase, metaphase, anaphase, and telophase. And you can see all the colors, the red and blue, and all the different types. And then here is the second one. This is the second phase where you can see. But there's also like one that is like a bad one. It's not like a bad thing, it's like an exemption to this like genetic information and all the meiosis steps, and it's called the non-disjunction. This is where a cell receives too many or too few chromosomes. In other words, non-disjunction is the failure of the chromosomes to separate, causing like the body to have abnormalism, maybe like Down syndrome. Like Down syndrome, that's an example of non-disjunction. But this is something that scientists are still investigating. They're still, because there's like so many of them. Like you can see sometimes you can go to the mall and see people that has like different type of structure, to say it like that. But that's the reason why, because the chromosomes fail to like separate correctly, and then it happened like a not normal distribution, and then they look like that. But yeah, guys, I hope you understand a little bit. I know my English is not that good because I'm still, I have the accent. But thank you for watching, and I hope you like it. Have a good one.